It is a pleasure to introduce Professor Santiago Elena for this webinar. So let me start with a brief sketch of the Santiago Elena profile. Santiago is a research professor at the National Spanish Research Council at CSIC. He is also an external professor at the Santa Fe Institute that probably most of you know. This is one of the leading world centers in complex systems. He is also adjunct professor at the Chinese Academy of Agricultural Sciences Institute of Plant Protection. And he was elected as EMBO member in 2011. And he has been also recently elected as a member of the American Academy of Arts and Sciences, just this year, 2020. He's also editorial advisor for journals on, for example, the BMC Evolutionary Biology, and he is also a co-editor-in-chief for Virus Evolution, a journal from Oxford University. And Santiago's research is focused on the evolutionary biology of viruses. He's very interested in study this evolution of viruses within the framework of population genetics. And also related to this is exploring the mechanisms that generate and maintain genetic variability, focusing on RNA viruses. He's leading the Evolutionary Systems Virology Group at the Institute for Integrative Systems Biology in Valencia. And today he will talk us about why some patients develop a severe syndrome while others remain asymptomatic, early warning signals associated with the phase transition from health to disease. As Albert was saying, the research of Professor Elena is mainly focused on experimental evolution, but he has been collaborating and doing very outstanding research in complex systems and also in mathematical biology and dynamical systems. That is, he has been working, and the talk today, I think it's related also with complex networks and phase transitions. And this is just a brief introduction for all of you to know a little bit about his research. So Santiago, please, you can start when you want.
Okay, thank you, Josep. Thank you, Albert. First, for the invitation to this webinar. For many of us, this will be the first time that I give a conference, a seminar in this format. So I hope things will work okay. It's going to be a bit weird not to have audience in person, face-to-face, but I don't know how to handle if you have questions. Probably you can just click on the raise your hand icon in the bottom, and we will try to answer interactively. So let's try to make it as similar as a face-to-face conference. So let me first turn on my presentation. Okay, I guess you should be seeing a full screen presentation now. That's correct, Josep? You see that?
Yes.
Okay.
It's okay.
Good. So we are on schedule. We are working. So as Josep said, we are working on the Institute of Systems Biology in Valencia, which is a mixed center between CSIC and the University of Valencia. We are working here for now. I don't know what will happen after October, but now we are here. And my talk today is going to be a bit weird in several aspects. First is that because the COVID and the webinar complex system series is about COVID, I have been asked to talk a little bit about COVID. But we are just starting working experimentally in the lab with COVID, so I have no results to report so far on COVID, but I will try to present some of our research with other viruses that can be applied also to COVID as data start accumulating, and I hope you will at least get the idea of what we try to do, and perhaps even if you get interested, collaborate in the future. Oops. So, okay, here. So COVID, this is an emerging disease. You know what is an emerging disease? It's a disease that is either appearing from the first time, like COVID, SARS coronavirus 2, or a disease that is periodically reappearing in populations. as for instance influenza. Once in a while we get a new combination of the segments, and then that means it's a new virus. It's still influenza, but it's a new virus. It's considered a new virus. This is data from yesterday, just to illustrate how the epidemic is going. It is really growing too fast. It was kind of growing very fast and then stabilized for a while, and now it's still, notice that this is in log scale, so this is growing exponentially right now. Here you have the numbers. Spain, which is doing very badly according to the some politicians, turns out not to be the first one, not even considering the population, although they keep claiming that we are doing worse than nobody else. So one of the things about COVID that people realized almost immediately was that the disease develops in different manners depending on several factors. Some of these factors were like age, so elder people get sick and unfortunately reaches a very severe disease and dies, whereas younger people doesn't. Males seems to be more susceptible than females, and there is also some other factors that we still don't understand on why some people is really developing severe symptoms while other people is just asymptomatic. And this is very important to understand, because not only because we can prepare or develop control strategies, not only therapies, but also vaccination strategies, but also because I mean, we need to know which genes in the human population are related to resistant to disease, and that will be important. Oops, again. So disease, just for the sake of having some background, the disease, the clinicians classify the disease into 3 phases. The phase 1 is when the virus gets into your body and usually cause a mild symptom disease that usually is confused. It can be confounded with a cold or flu. This is basically what happened at the beginning of the epidemic. Probably the first flu coronavirus disease cases were in January, as early as January in Europe, but they were simply classified as common flu. Phase 2 is the pulmonary phase, so where the respiratory syndromes start to appear, the symptoms, people get persistent coughing, they are short breathing, there is very low levels of oxygen in blood, and in some cases even blood clotting. This immune system responds very strongly to this infection, to this massive infection in the lower level of the lungs, and this is the phase 3, which is the hyperinflammatory phase. Basically, it's the immune system that becomes completely crazy. It's what is called by immunologists, is called the cytokine storm, which basically is your own immune system that is attacking all the tissues of the. I mean, it's a kind of a autoimmune disease. No, the immune system is attacking all the tissues where the virus has been previously present, and this is what is actually causing most of the death cases in elder people. No, the autoimmune response of the immune system. The big question for us is, how can we predict what drives the jumps between these phases? So if we know something about each individual host, each individual patient, can we predict whether the patient is going to move from phase 1 to phase 2 and then stay there, or moving from phase 2 to 3 and then stay and then develop a very severe thing, or whether the situation is reversible? So once you reach phase 2, to which point within phase 2 you can move before the situation is completely reversible? Okay, this is the type of questions that we would like to address. So the first thing we have to understand is that disease is a network property. Here, what I'm showing you is basically two examples of two viruses, very quite different viruses. One is human influenza, or yeah, I would say HIV, the AIDS virus, okay? And red balls represent the viral proteins, and blue dots represent human proteins. And this is a protein-protein interaction network, experimentally determined, where it shows that each one of the viral proteins is interacting with a bunch of different factors in the cell, and from these interactions, the disease, the symptoms develop, okay? If this GP120 is kidnapping these proteins, probably this has an effect on the interaction that these proteins have with other proteins, and then that will transmit the effect of this, the direct effect on this protein will be transmitted indirectly to other proteins in the proteome. This is another case. This is coming from our own research in the lab. This is for a plant virus. It's called tobacco H virus. Actually, it's a virus quite similar to the corona in genome structure. It's much smaller. You know that, probably you already know that corona is the biggest RNA virus that has been identified so far. But TB is a more classic, small 10 kbs virus. It encodes for just 10 proteins, and it does a very variable disease in the host plants. This is just to illustrate two proteins for which we experimentally determine all the interactors that these two viral proteins have on the host, in this case, tobacco plants. The red arrows represent direct interactors, okay? So proteins that physically interact with these viral proteins, whereas blue arrows are one-step interactors. So that means that they are not directly interacting with the viral proteins, but they are interacting with the interactors of the viral protein. We have been doing some modeling of the networks, and we show that in six interactions, the entire interactome of the plant is already affected by the perturbation of the virus, okay? So this is just to show you that the development of the disease depends on a very complex number of interactions between viral proteins and host proteins in every system. It doesn't matter if it is a human virus, or it is a plant virus, or if it is a bacteriophage. The situation will be the same everywhere. These genes that are directly interacting with viral factors, we usually call virus-regulated genes. This is just a question of terminology. You know that biologists, we like to give names to everything. So we call these first-order interactors virus-regulated genes. So this is probably very naive. From now on, the next few slides will be a very naive presentation of what is a phase transition and what is a complex system. And it's more, I have this slide because usually I give this type of seminars to biologists, and for many of them, this is kind of new stuff. So I'm apologizing to those of you that are more on the physics, mathematics side, and you are very familiar with all these ideas. So what is a phase transition? Basically, is represented by these diagrams here. This is a smooth transition. In biology will be, for instance, the decay of a protein. So with time, the parameter you are measuring is just slow in decaying, but in some situations, you can see a sharp decline in the situation. So, for instance, in enzymology, this is a typical Michaelis-Menten or Hill function, where you have a sudden decline in some property that you are measuring, but you have more extreme situations like this, where you have two different stability points, and they might be stable or unstable, depending on the mathematical properties of the system. Okay, so basically, what is represented in this panel is a system which is poorly connected, and where you stress the system the system responds in a kind of linear manner, something like that. And this is the properties of such a system is modularity and heterogeneity, so you can define modules that are not connected, usually it's a smooth system. Whereas when you have a very complex system like this one represented here, which can be, for instance, a metabolic network or a gene regulatory network, you have lots of connections between elements and they are highly connected, and that means that this is a very robust system to random attacks. So you can remove one of these links or one of these balls and the system is still stable, it's working fine. But at some point, if you attack the hubs or the bottleneck nodes, then there is a sudden transition in the system where basically you go from one state that is metastable to another state which is also metastable. Okay, so systems that are highly connected and homogeneous usually are robust to perturbations to random perturbations, but they are very sensitive to selective perturbations. Okay, it's basically like the internet or the airports network in the world. There are hubs. If you cancel the traffic through Heathrow, probably all Europe will be affected. Whereas if you cancel the traffic in, I don't know, in Valencia, probably many things, nobody will care at all about that. The system will still be working. Okay, so these are two ways that the potentially that molecular biological systems can organize in a very sparse modular way or in a very highly connected molecular way. So, as I say, disease is a network property. This is a figure that I took from this paper, published almost a decade ago by a group of Chinese computational biologists. And basically, it's illustrated the idea that you might see as the systems, the cells, or the cells, they are in an homeostatic state. So it's functioning perfectly well. You have different genes or proteins or metabolites. You can imagine these balls as any element you want to consider. They are just elements in a network, and they keep a certain concentration level, which is in equilibrium, and they react one with another, or they interact with another, represented with these blue lines. Okay, this is a stable state, which is here represented as a potential landscape, in a minimum of the potential landscape, and this is a stable system. That will be our cells in normal, stable conditions. What happens if we induce a disease? If the system suddenly is perturbed to a certain level, either by internal perturbations, let's say, for instance, cancer, or genetic syndromes, or poisoning, something like that, or externally perturbed, for instance, by pathogens, or by I don't know, some chemicals that you can get into your body, like drugs or things like that. Okay, the system, the potential landscape probably changes because you have changed the conditions of the system, and you get into what is called a pre-disease state, which is still reversible in the sense that if you remove the perturbation, the system might evolve back to the situation, or might evolve to another situation. In this pre-state disease system, what you have is that some of the components of the system start to fluctuate among them, or their fluctuations in concentration are correlated, whereas they are disconnected now from the rest of the elements in the network. For instance, here I just representing three of the components start fluctuating in a correlated manner and disconnecting from the rest, and that means that the concentration of C1, C2, C3 might change dramatically from one situation to another as time moves on. Of course, as I said, this is a reversible state. So if you make a action on the system to reduce the perturbation, the system might well get back into the normal state. If you think in a drug, for instance, if you provide an anti, or if you think in a poison thing, you're gonna add the anti-poison, the anti-venom, and then the system might go back. If you have an infection by a bacteria, and then it's starting to perturb the system, and then you add antibiotics, the system might go back to the normal state. But what happens if the system is pushed to a new situation where it's not reversible anymore? You just change completely the dynamics of the system. You get into another potential function where the system just now moves and is completely different. And this is irreversible, and this is what is called the disease state by these authors, and in general by others. Okay, so basically what you have is a new situation in the system where the components might, as represented here, might still be connected, but with other concentration, or they might even be disconnected. Something that was previously connected now is disconnected, and this is an irreversible situation. So we can somehow call these things here, or the drivers of what molecules in the system are really being responsible of this shift between the normal, the pre-disease, and the disease. We can call them the early warnings. Okay, this zeta 1, zeta 2, zeta 3, if you are able to quantify their concentrations, and then suddenly you see that the concentration of these three molecules is suddenly fluctuating out of the standard normal concentration, before the systems go beyond that, you can already say that this is an early warning of the phase transition to the disease state. Okay, so that will be the idea. So this is these authors, they call this subnetwork. They call this subnetwork here responsible of elements responsible of the switch of this stability of the system. They call the dynamical network biomarker, and dynamical network biomarkers will be the early warning signal for the phase transition in complex diseases. So this is a toy model that they develop in their paper, in the supplementary materials of the paper. And one of my collaborators here further expanded, and we co-supervise a TFG, a trabajo fin de grado, of a physics student that proved some of the things in the model that were not completely proved before. So basically, you have a vector that determines the concentration of all the elements at a given time as a function of what was in the previous time plus the perturbation. And this is an n-dimensional vector. It's as big as you want. It can be the entire human interactome, or it can be the entire tobacco plant's interactome, protein-protein interaction, or gene regulatory networks. It's whatever network that might represent the physiology of the cell. Okay, and then the perturbation is an s-dimensional vector, which means it's the effect, the external effects on each one of the components of the network. Okay. So I'm not going to get into the math details. It's quite interesting problem from a mathematical perspective, and is somehow developed in this paper and a subsequent number of papers published by this group. But if you are more on the mathematical side, it will be interesting if you want to get into this problem. So, of course, there exists an equilibrium, a fixed point, in which the system is at equilibrium. Okay, you can calculate what is the value of the perturbation that brings the system to that equilibrium, and there are certain conditions that have to be met to make the system stable. And they, as I say, they prove those conditions in the paper. Basically, if these conditions are completely fulfilled, the system goes through a transition, a phase transition, when the perturbation parameter equals the PC value, this thing. Okay, so basically the system is under a kind of metastable situation until reaching until meeting these conditions, and then you get the sudden phase transition into another phase, which in this case will be the disease phase. So it can be shown, as they did, that this equilibrium condition is called the DMB, is the number of genes that determine, or are more important to determine the transition between the two systems. And these genes, the DMB, they are characterized by a number of properties. First is that the variance of the elements within the network increases. The covariance between the elements of the network increases, whereas the covariance between any element in the DMB and outside the DMB decreases. So as I said before, you have a subnetwork that starts co-fluctuating and they break the synchrony with the rest of the network. This subnetwork will correspond to the DMB, whereas they is now disconnected from the rest of the elements in the network. And finally, they came out with a statistical measure of this to quantify how strong is the effect of the perturbation on the DMB, which is basically this index down here that is composed by the average variance in expression for elements in the DMB. The subindex D means the gene belongs to the DMB, so it's the variance in expression for those genes times the correlation coefficient between elements that belong to the DMB, in this case these two guys here in orange, divided by the average in absolute value of the correlation coefficient between elements outside the DMB. Okay, so this is just to make the number bigger, as you will see in the next figure. So you can just play with this very simple toy model and write the differential equation that describe the dynamics of the system. This is the numbers here just are simply values to make the solutions of the system easy. I mean, to provide clean numbers and not decimal values or things like that. These are basically arbitrary numbers. So if you get the system and you can evolve the system as you perturbate it, and then you get something like that. So this PCC panel represents the Pearson correlation coefficient between pairs of elements in the network. And so as you see, there is a critical P, the perturbation parameter, where basically the variance of some of the element within some of the pairs increases, whereas the variance of other pairs decreases. The standard deviation for the elements, you see that some of them don't change. This will not be part of the DMB, whereas these two guys that increase suddenly in variance will be C1, C2, which are the two elements in the DMB. Okay, so you can do just numerical analysis of this small toy model, and then you find that there is a bifurcation exactly when there is a sudden increase in variance between the elements inside the DMB and a decrease in the correlation between between elements in the network and outside the network. I mean, sorry, the DMB and outside the DMB. These are two examples from real data that they add in the paper. They are not related with viruses. They are just a chemical injury. If you breathe phosgene, which is a pesticide, then you get a lung injury. And you see here, this is for humans, so this is based on the protein-protein interaction for humans. So this is basically the concentration of the phosgene, the strength of the perturbation, and this is how the standard, the variance basically increases for treated patients and non-treated patients. The correlation coefficient, don't remember what is this, and this is the composite index. This is the number I showed you before down here, okay? The composite index. So you see that there is actually a maximum when the perturbation really becomes irreversible. This is another example, is the chronic the development of liver cancer when you have a chronic infection with hepatitis B virus. This is more related with viruses, but again, it's the same. How you see that a certain stage on the progression of the disease, what the clinicians call stage 3, you see that the standard deviation increases, partial correlation, Pearson correlation coefficient increases, and then the composite index increases. So basically, when the disease reaches stage 3, there is no way back, there is no reversion, and the system suddenly suffers the sudden transition from the reversible, almost healthy stages to the non-reversible, healthy, unhealthy, and disease stage. This other way of representing, as I say, this is the same data. This is for the cancer thing. Is basically put in the context of the entire protein-protein interaction network. In humans, as you can see here, they managed to draw the network separating the elements of the DMB to highlight that they are behaving in a different manner. So basically, you see, this is just an amplification of the same. You see that these things start with as the disease progresses. You see how at the beginning the number of red and green balls were almost homogeneously distributed along the entire network, and suddenly you start seeing an increase in the number of correlations of elements within the DMB. So this is the DMB, and this is a detail of the DMB. You see how the correlation between elements just keep growing until the phase transition happens, and then you reach a new equilibrium state, which is the disease itself. So the system will be reversible here in these early stages, but once you go through this phase, then the system becomes absolutely reversible, and that will be the disease. And these genes that start fluctuating together, they are what we call we are now calling the DMBs, the dynamical network biomarkers, and these are indicative of the early warning. So if you analyze the gene expression of a patient that is suffering cancer, colorectal cancer, and you're seeing that the expression of these genes in red here starts fluctuating in a tightly correlated manner among them, but not anymore with the rest of interactors, then you can predict that the sudden transition to disease is almost ready to happen. So there is an algorithm, although they never implemented a computer algorithm, so there is no, unfortunately for us as biologists, there is no bioconductor package that can do this analysis. So you can basically implement your own pipelines just following the indication. There is a modification of that, which is what we have done. And basically, what we use as data for this analysis is gene expression data, is a microarray or RNA-seq data. I don't know how familiar you guys are with all these high throughput gene expression techniques in biology. It means that you know what I'm talking about. Means that you can measure the expression of all the genes in a given time point for your tissue, cell, or patients. Okay, so basically you just measure all the genes, you analyze the gene expression of the of the disease or the target patient versus the control patients, and then you see which genes are really changing, differentially expressing in infected, non-infected, or poisoned, non-poisoned, sick, non-sick, whatever. You see the genes that are differentially expressed, then represented here by different colors, cluster the genes according to the expression and time of sampling, and then based on the correlation coefficients, you can determine which genes are fluctuating in a correlated manner with time and suddenly changing the correlation with the other genes in the network. That will be an indication that these are the DMBs, the dynamical network biomarkers. And then you can analyze what genes are there and what they do in terms of function. So, but basically this is kind of a brute force algorithm. So just correlation coefficients among n and see when you start seeing changes in the patterns of correlations. And then you can basically go to the next step, treat those as candidates for members of the DMB, and then calculate correlation between them and outside, and then finally calculate the index that I mentioned before. This is a little modification in the index, simply to avoid dividing by 0. This can be an arbitrarily small quantity, just simply to avoid dividing by 0. The bad thing about this format of presentation is that I don't know if you have questions or how your faces look like. No? Sometimes when you give a seminar, you look at the faces and you see, okay, things are going well because people seems to be paying attention, or people is just sleeping. So, but now I have no clue on how you are receiving all this. Anyhow, I'll keep moving. So more recently, the same authors have developed a modified version of the algorithm that takes into account noise, very noisy data. This is just a case with a small noise, and this is a case with high noise. I mean, obviously, the more noise you have in your data, in your time series data, the more difficult it is to infer what are the dynamical network biomarkers. This is just the expression data for a given. Imagine that this is gene expression for one particular gene, and then you see a sudden transition. If it is not noisy, where the sudden transition happens, it's actually quite easy to determine. There is an autocorrelation, temporal autocorrelation that also changes. Whereas if you have a big noise, then you have a certain uncertainty where actually the phase transition is happening. These authors, they came with a way of transforming the noisy data to get better estimates of where the transition happens. Okay, so they have been keeping working on the problem. Again, in this paper, they develop an improvement of the model. There is another improvement of the model. They use what they call state transition-based local network entropy, SNE, which basically allows you to better understand what are the DMBs, the dynamical network biomarkers, because they allow you to define what are the real DMBs, which here are indicated in this orange reddish circle. This will be the real DMB, whereas other things like this light blue will be things that are not strictly belonging to the to the core of the DMB, but they belong to kind of a boundary of the DMB. So they are directly associated with the DMB, but they are not part of the DMB. Okay, they keep fluctuating with the non-DMB members. Okay, so they keep still in good relation with the outside, although they are directly affected by the members of the DMB. Okay, and this is based on these transition diagrams, and you can calculate the probabilities from moving to one state to another. It's kind of a Boltzmann distribution where you have all the possible stages, and then you calculate probabilities for all of this. The data here again are the network model, and this is for humans. There are several network models, and for model plants we also have several network models, and then the expression data. So the size of this, the network model gives you the edges between nodes, and the experimental transcriptomics gives you the size of the ball. So how much of this ball you have in a given time point. And here is the big jump. I was talking about coronavirus, then I give you an overview of the theory that we are trying to implement in our work, and now is the model system we have been using in the lab to test this theory, the application of this theory to a real viral infection. Before moving to this to the Systems Biology Institute, I was in a plant molecular biology institute with a strong component on virology, and we use a plant virus as model system. Okay, Plant viruses, they have certain advantages over animal viruses, and you can imagine the most obvious one is that you don't need to worry about getting yourself infected. Now that we are coronavirologists, because we already work with coronavirus, you have to worry more about protecting yourself from infection than here with the plants, that basically you don't care about plants getting infected. So our experimental system was the tobacco H virus. This is an electron microscopy of the virus. Each one of these sticks, here you have 3, here you have 2, here you have 2. Each one of these sticks is a viral particle, how they accumulate on the cytoplasm of the cell. This is a schematic representation of the particle. These blue elements are the capsid elements, and there is an RNA molecule inside. This is the structure of the RNA for those of you that might be more interested in biology. It's just a single RNA molecule of approximately 10,000 nucleotides, 10 kbs, that encodes for a single very long protein, which is, one is translated, itself folds in a given structure and then chops itself into the mature peptide. The chopping is made by proteases. P1 chops here, HCC-Pro chops here, and then NIA-Pro, the major protease, chops all the rest of the elements. Important proteins here, all are important, but this is the code protein, the green one here. This is the replicase, is the protein that copies the RNA genome into the progeny, and this is the viral protein G, which is basically protecting the 5' end of the RNA for degradation by the cell. This virus, as I say, is an RNA virus. It belongs to the potivirus family, potivirus genus of the Potiviridae family, which is part of the picorna-like superfamily. Picorna-likes are hepatitis C, are poliovirus, are foot and mouth disease virus, and other viruses that have basically the same genome structure and the same way of processing information. Okay, so this is a very common structure for viruses in nature. It's a plant virus, but still is very informative about how other viruses do, how other viruses do their whatever they do inside the cells. And this is just to illustrate the symptoms. This is a healthy tobacco plant, so it's very green, nice. Where this is a sick plant, where you see the leaves are becoming yellow, and both plants have the same age. And you can see that this is substantially smaller than this. And if you keep the plants for longer, this plant will grow as big as a tree, couple of meters high, where this will never be higher than 50 centimeters. And the leaves at the end, these yellow things became brown, and then the leaves die and the plant dies. Okay, so that's our experimental system. We have been working a lot, as I say, on the molecular biology of this virus, and especially on understanding or inferring the protein-protein interaction network of the virus and the host. So we first characterized what were the interactions between viral proteins themselves. So NIA-B interact with VPG, with NIA-Pro, and so on. And also we characterized the cellular interactors of the viral protein. So we know which viral proteins are more connected with this host than others. It has been a number of papers working out these things. VPG is highly connected with lots of elements, and that makes sense because it's involved in translation of the RNA into proteins, and it's also involved in the transcription of the RNA into the intermediaries of replication. Whereas NIB, this is the replicase gene that interacts with some cellular factors like transcription factors and things like that. And this is the suppressor of RNA silencing, is the major so plants defend of infection, and the virus blocks the defense using this protein. So it interacts probably with immunity things, all these immunity elements on the plant cells. Okay, and this is blocking the immunity response. So we have been, as I said, we have been working for many years on this system, and we know quite well how the things evolve, how the disease progresses. And this figure which is apparently simple, actually contains a lot of information that I need to describe you carefully. So here, we are plotting on the y-axis is the fitness of the virus relative to the wild type. And what I'm plotting on the x-axis are different viral genotypes, okay? So these are the names of the viral genotypes. They are named this way because they have mutations in certain proteins. So in the CI, in IB, HCC Pro, and so on, they are mutants on those proteins. And as you can see, some mutations have a stronger impact in fitness than others. So this is the wild-type virus, the non-mutated virus. So for instance, this mutation represents almost a 15% reduction in fitness, whereas this mutation here just represents barely a kind of 10%, or this one represents almost a 40%, and these ones represent an 80% effect on fitness. So these viruses, these mutant viruses, accumulate in the plants in a different manner. Okay, and they produce symptoms that are actually different. So these numbers inside is a measure, is a almost qualitative, semi-quantitative measure of the intensity of symptoms. If 5 represents the symptoms that I showed you, that I showed you before in this slide, this is wild-type virus. If these symptoms are represented by 5, 4 means weaker symptoms, 3 means even weaker, and 1 means almost no symptoms. Okay, so there is a correlation, of course, between viral replication or fitness and symptomatology. We have been doing a lot of transcriptomic analysis of these mutants, so we know precisely how the gene expression profile of the cells depends on the fitness of the virus, the replication of the virus, and this is represented by this dendrogram here. So the dendrogram is telling us that these two viruses produce this similar alterations on the profile of gene expression of the plant. Okay, so if you look at the magnitude of the perturbation in gene expression, these two are more similar, these two are more similar, then these 3 are more similar among them, these 3 are more similar among them, then they cluster together, and finally, this virus, that is very weak virus that produces almost no symptoms, is the less similar to the other in terms of perturbation to the transcriptome of the virus, to the gene expression of the host. Sorry. So we can classify, we can define disease stages from almost no disease, which will correspond to this virus, that is not perturbating the genome, the transcriptome of the cells, and is producing very mild infections. That will be phase 2, phase 3, 4, 5, 6, and 7, where phase 7 is the very strong symptoms, lot of viral accumulation, and a strong perturbation in the transcriptomic profiles of the plants. Okay. All this data, the discussion of everything here is already was published a couple of years ago in MBE, Molecular Biology and Evolution, and it's there if you want to take a look at it's more precisely what the data looks like and what we do with those data. So the first thing is to try to apply the DMB theory to this data. We have phases in the development of the disease, and the question is at where the irreversible transition happens. Okay, at which stage in the disease the plants are cannot recover anymore. These plants, as I say, these plants are very mild infections. They grow mostly well. These plants are very, very sick. They tend to die after a number of days. So in which point of the disease progression can we consider that the phase transition happens from the reversible to the irreversible metastable situations? So the first to do, so as I say, this is the algorithm, as I say, is just looking at correlations and then trying to define what genes are changing the pattern of correlation along the disease progression. That was brute force, that was too much for us, so we came up with the idea of trying to simplify things. So first, as a first preliminary step, what we did was looking at the shape of the expression profile along the different disease stages. What do I mean by that? So we took a gene out of the 28,000 genes in tobacco. We took a gene and then we looked how was the gene expression along time. And you have different possibilities. You have the possibility of not change at all. You have the possibility of a sudden decrease, or you have the possibility of a sudden increase. And either the increase could happen very early on time, at intermediate categories, or very late on time. Okay, or you may have a smooth increase or a smooth decrease. Okay, there are different possibilities. But basically, we counted the number of biphasic profiles at each one of the disease stages. By doing that, what we're looking is a gene that was expressing high and suddenly dropping in expression, or vice versa, a gene that was very low in expression and suddenly increased in expression. Okay, and we did that for the 28,000 genes in the plant. And this is the counting of genes for each one of the disease stages. So you see that at various early disease stages, there is still a number of genes that show this sudden change in expression, like 500 something, or 600 here. And then suddenly, as we get to between phase 3, phase 4, we see a sudden jump to thousands of genes that they change the pattern in a very sudden manner around this category, and then back to a few genes changing the pattern later on the disease. So this is kind of suggesting that between these categories, around category 4 of the disease, something is happening in the plant that is kind of a sudden transition in a lot of genes at the same time. So then we decide to call these early transition genes, intermediate transition genes, and late transition genes. And then we analyze each one of these sets of genes on the next step. So let's say which genes are here, which genes are suffering such transition, early, intermediate, or late. So in green, just a few examples. I'm not going to enter into details of which genes are doing what, because this is more for plant pathologists than for the audience today probably, but just to show you that things kind of make sense. So green means genes that increase in expression, red means genes that decrease in expression in a sudden manner. Okay? The top genes are ethylene response, which is the hub regulator in response against pathogen. This was well known, and this was a kind of confirmation that what we were obtaining was not crazy results, but something that made sense. If you find a well-known regulator of the response to infection as the top gene, that makes sense. Okay, or genes that are also response to what is called in plants the pathogen-related family of proteins. Those are the first. This is kind of the regulator of all the system, and this is kind of the first layer of defense of the plant against infection. This is inducing a lot of other things. What is going down? The catabolism of sugar and starch. So, for instance, if the plant has to start moving, plants can either grow or defend. They cannot divert resources to both function at the same time. If they decide to defend themselves, then it's at the cost of not growing. Okay, so that kind of makes sense and motivates us to continue. That was about the early. About the intermediate, what you have is things like activity of the mitochondria, which is these are kind of the metabolism of the cell start being affected, the production of materials, they start going down. Things that are known to be actually needed to respond to infection are activated. So things make sense from a plant pathology perspective. They were not surprising, which is okay. And what happened at the late stage is basically things that has to do with metabolism, or they have to do with the growth of the plant. okay, actually with plant growing. And it makes sense. The plant tries to compensate the syndrome, the disease, by trying to activate genes that produce tolerance to infection and allow the plant to grow even when infected. Okay, things make sense. So far, so good. So now we try to identify the DMBs. Okay, so far we have classified genes in those suffering a biphasic change in expression at 3 stages: early, intermediate, and late. So now we took those genes and then we incorporate the information of the network. Okay, we have 2 network models. We decide to work with 2 different network models. One is the protein-protein interaction network for tobacco, and one is the transcription regulatory network. Okay, so basically, I'm going to show you the results in parallel for the protein-protein interaction and for the transcription regulatory network. This is the I index that I showed you before from the introductory theory slides. And what you can see is that if you, for every one of the genes in the, I mean, how these things work. Remember, we have a gene, and then we know is connected with whom is connected in the network. And we know that they are suffering as a sudden transition that is coupled, okay? So we know that they are correlated in the change on expressions. So we can just start looking at the network by looking around those genes and see at which point they stop being correlated. So, and then we decide that these are outside those are outside the DMB. So there are several DMB candidates. Here they are in different colors. So one candidate will be switching very early and then go down and then get back in expression. Is this black here. Others are like this pink one, is just has a maximum here, and others they have a maximum later on time. So there are several candidates of DMBs from the protein-protein interaction network. For the transcriptional regulatory network, there are less candidates. There are a few, and they are strongly associated with phase 4 of the disease development, is where we see the maximum number of the maximum I values, okay, for this candidate here or for this candidate here. If you look at the network where they are, this is what is called the IA1, the protein-protein interaction network. This is a public network you can download from databases. It's a very sparse, not completely connected network. So these are different modules in the network that are connected, but they are disconnected among them. And in yellow, what I'm showing in yellow are those genes that are part of these DMBs here, okay? So this DMB, and so on. So you see that some of them cluster into modules, like this module here that contains a number of yellows, or this one here in the core of this module, you have some DMBs. Some of them are more disconnected, some just have an interactor or two, but some are really well connected in the major component of this network. When you look at the transcriptional, no, we keep on that. So what things are here, for instance? What type of genes we have in this network or in this network? We can go network module by module and then see what functions are being affected by these DMBs. So we have 27 significant DMBs for the protein-protein interaction network. I'm just going to mention a couple of them. DMB 134 is the larger in size. It's the one that contains more elements, and among those elements are transcriptional coactivators that include heat shock proteins. Heat shock proteins are well known to be involved in response to every kind of stress. So I will say that if we are looking at the transcriptome of the plant, and then you see that heat shock proteins suddenly start fluctuating in abundance, that will be a clear indicator that we are getting close to the phase transition. And the triallose phosphate synthase controls the synthesis of triallose, the sugar that is an intermediate it's a signaling molecule for the response to pathogens, not for viruses, not precisely for viruses, for fungi or bacteria. Okay, and it is a suppressor of this defense response. Okay, so basically, if you see that these two things start fluctuating, then you can predict that the system is going to suffer the transition from the pre-disease to the disease stage. This is the second DMB, and it contains things like proteins involved in the assembly of the nucleosome, also related with heat shock proteins, and also transporters of ATP, so energy basically. Okay, you see that the energy, there is a tremendous export of energy from the chloroplast to the cytoplasm that is out of normal. That means that probably the system is getting close to the transition to the disease. Wait a second, where is this? Oops, I'm missing a slide, I guess. Yeah, I don't know where it's at. One slide is missing. I'm sorry. So well, maybe it comes later. So what happened if we look at the transcriptional regulatory network? The two previous slides were for the protein-protein interaction network. This is for the transcriptional regulatory network. We have 19 significant DMBs. By the way, the significance is evaluated using bootstrapping and things like that. So we just resample the network and decide and reconstruct the DMBs for the random samples and see which ones are significant and which ones just get by chance. So there are a total of 19, just to mention a couple of the largest ones inside. The number 34 is involving proteins that are, a priori, we will not assign a function to some of the proteins. They are just proteins involved in metabolism, but we don't know how to relate them with infection. Some are related with some of the stressors like salt, growth of the plant, some transcription factors, but they're not easy to see a pattern. No? They are just things that tell you that the transition is going to happen, but it's not easy to see why the transition is associated to these genes. The other component is the 36, okay, and then you have, again, here is a bit more easy because they are related with response to infections or to herbivory, so chemical signalings and responses to abiotic stresses. So, but yeah, a priori, we will never predict that these things will be early warnings of what is going to happen later on time on the progression of disease. The DMB40, you have a bunch of things. This is, but again, it's basically the same idea. You have things that some are easy to understand, some are not that easy to understand in terms of what you will predict. But the point is that when these things start, they fluctuate together, and then they start losing their connection with other members in the network, and that means that that's going to indicate a change in the stability of the system and the transition to the disease. And here, this slide should be early. So the DMB, the TRN DMB36 and the 40, which are this and this. If you look in the context of the transcription and regulatory network, which is highly dense, they are actually quite, this is quite well connected. They all belong to the same modulus. You calculate the modularity. Actually, this is a significant module, where this is not a significant module, so this seems to belong to different modules in the network, but they are connected. It makes sense that when these fluctuate because they are connected and so on. What else? So the final 3 slides is trying to get some more ideas about the physical property, the network properties of these DMBs. So what I'm showing this slide is the entire transcriptional regulatory network for, this is for Arabidopsis thaliana, not for tobacco. but we can always use the ortholog genes of tobacco and assume that they have the same connectors, the same interactor than in Arabidopsis, and then use that Arabidopsis network to analyze the tobacco. Okay, this is to put a note of caution with what is coming now, but because we are extrapolating something from Arabidopsis to tobacco. But it's the best thing we can do so far. So this is the transcriptional regulatory network of the entire transcriptional regulatory network of Arabidopsis. 24,000 genes are here. This is something that we published a number of years ago. And this is just to show you some of the two properties of this network, is that the incoming connectivity is kind of log linear, whereas the outgoing connectivity for the transcription factors is log-log linear. So it's kind of a power law here. And actually, this is what you expect for most of the biological networks. They are small scale free, power laws, and so on. We can do an experimental thought, a mental experiment, which we published this experiment. This is a complete mental experiment we published in an opinion paper a number of years ago also, in which we basically predict what will be the power law, the effect on the power law distribution if the viruses basically interact with random set of genes in the interactor, with genes that are poorly connected or with genes that are highly connected. Okay? Imagine that in red, what I have here is the actual log-log or degree distribution in log-log scale, the degree distribution for the entire network in red. Okay, this is the network in red, and this is the power law distribution. Okay, for that network. Imagine that now we sample a number of genes completely randomly from the network. If you do that and you plot what will be the connectivity distribution for that random set of genes, you will get the same line here, plus minus some fluctuation, but will be something that will fit on the same line, because it's a random sample of a small wall scale-free network. What will happen if instead of sampling at random, you specifically sample genes that are poorly connected, like those marginals here? If you do that, then you see an enrichment in poorly connected genes and a reduction in the number of highly connected genes. So your power law will look more like this. It will be steeper, okay? It will have a more negative slope, but than the null model, than the red one. In the opposite case, what happens if you, the virus in this case, randomly, no, not randomly, but specifically picks nodes that are highly connected in the network, like hubs or bottlenecks in the network. If the virus specifically picks genes that are essential in the network, hubs or bottlenecks, then what you will have is something like this situation here, where the power law now is flatter. You have less poorly connected genes than expected, and more highly connected genes than expected by the null model. Okay, that was our thought, our mental experiment. And then we tested that with a lot of data in this paper, and actually this is the case. In this paper and references therein, that is the case. The viruses tend to more selectively interact with hub genes. When you do that calculation for the entire protein-protein interaction or the entire transcriptional regulatory network, and for the subsets of DMBs, for the elements of the DMB, okay, you have here the data. This is for the entire protein-protein interaction. This is for the DMB. This is for the entire transcriptional regulatory network. And this is for the DMB obtained from the transcriptional regulatory network. You see that things change. For instance, the shortest path is significantly larger. Asterisk means that they are significantly larger in a t-test. Okay, this is larger for both the PPIN and the TRN. So DMB seems to be, the shorted path seems to be slightly larger. The neighbor connectivity is much shorter for the protein-protein interaction network, but more they are larger for the DMB. But what is more interesting in the light of this previous slide is to look at the slope. The DMBs, they have significantly flatter slopes than the entire networks. Okay, significantly flatter slopes means a situation like this. So basically, this analysis is telling us that the DMBs that we have inferred are actually genes that are highly connected in the corresponding networks, either the protein-protein or the transcription and regulatory networks. So they are hubs or bottlenecks in the network. So the virus, so basically this is consistent with what we knew before about the viruses specifically targeting genes that are highly connected, hubs or bottlenecks. So basically, this is telling us that the DMBs are being directly affected by the interaction with the virus. And I think that's all I want to say. I don't know actually how the time is going. Yeah, probably have been talking for almost 1 hour. So I'm going just to stop here, acknowledging the people that did the experimental work in the laboratory, a number of postdocs and PhD students, and Juan Carlos is a master student that is a physics, TFG, trabajo fin de grado, and Adrián is a biotech trabajo fin de grado. They work together on this project. Juan Carlos did more the math side, Adrián more the bioinformatics side, and collaborators here at the Institute is José Ángel Oteo, which is a theoretical physicist that supervised Juan Carlos with me. And the money always came from the typical Ministerio, Generalitat, and CSIC. And that's all. I think that it's time for questions, if you have any. Okay.
Okay, Santiago, thank you so much for this really interesting talk, mixing multidisciplinary approach with experimental and complex system stuff. So for the moment, we do not have any question here in the chat. So if anybody has some question, just write it in the chat. Meanwhile, I will start with one question. Mm-hmm. So so the research on early warning signals and critical transitions in terms of diseases, I think this is a very recent and promising topic. And one thing that I don't know if you have thought about it or could be possible to do with your experiments or even with the models, is to know something about the timings between these transitions, whatever the control parameter is. So you have some kind of control parameter that makes your system to jump from one state to the other. But here, timings would be crucial, especially for infectious diseases.
Yeah. So as you say, the time here, particularly in the plant experimental system, is kind of discrete categories of disease that we define in terms of a bunch of different properties, no? The amount of perturbation in the transcriptome, the severity of the symptoms, the virus accumulation, and things like that. But you can use, and this is something we are now working in the lab, collaborating with the computer people in the institute, the computer scientists in the institute, is to analyze more detailed progression of disease. Like in databases, you have an amazing amount of transcriptomic data, for instance, for HIV, where they have been tracking patients day by day, and that you have the expression day by day. So you can really get, long time series. Long time series maybe 12 or 15 points. It's nothing compared to time series in ecology, where you have years of observations, or in physics, where you have years or very long time series. Here is about that. It's 15 days, for instance, looking every day at the expression of genes. In some cases, we also have cell cultures, and then we are taking samples every hour or things like that, but still we are on the same magnitude of days.
Okay, so we have a couple of questions. The first one is a little bit long, but I think I will have to read.
Can I see the question? Is any way I can read the questions?
They are in the chat. Yeah. So the first one by David Reguera.
Okay.
If you don't mind, I can read it. Okay.
Sí, for the rest.
Yeah, David says, you are considering a viral disease as a mere deregulation of the gene protein expression network, but sometimes the problem is just the virus killing the cell rather than deregulating specific gene or protein expression. And the inflammatory response may be similar to that caused by other non-viral diseases. So in terms of the disease, up to which point the DNBs of a viral disease are unique? And then, for instance, would be the DNBs of COVID-19 be different from those of a normal pneumonia?
Okay. Hi, David. Nice to talk to you. Good questions. I mean, what you say is that sometimes the problem is that the virus kills the cell, but the virus kills the cell because it's kidnapping the resources of the cell. It's not killing the cell by, I don't know, by entering there and, I don't know, asphyxiating the nucleolus with the hands or something like that. No, it's not like that. The virus kills the cell because it's using resources of the cell, because it's rewiring the metabolism of the cell, because it's blocking the expression of some essential cellular genes. So the cell dies because the combining effect of all these things. Okay, so it's still a network property. I mean, because it's somehow affecting the expression of multiple things in the cell. It's a bit more complicated because for us the network is like a big network of things, whereas in reality it's not like that. No, there is some genes that interact in the nucleus, some genes that might interact in the cytoplasm, some proteins that interact in the chloroplast or the mitochondria. So that's adding a level of complexity that, of course, we are ignoring. And your question about if the inflammatory response might be similar for viral infections than other non-viral diseases that also, for instance, autoimmune diseases, that's an excellent question. So that's an excellent question. And at this point, I don't know. I don't have an answer, but that will be something interesting to test. For instance, as you say at the end of your question, comparing the DNBs for COVID-19 and for influenza, for instance, that's in our pipeline to think of things to do. I never thought on pneumonia, on bacterial infection, but it would be also nice to compare virus and bacterial infections. At least in the plant systems, seems that fungi, bacteria, they share lots of responses. Viruses also share some of the signaling, but not all. So I would say, I mean, that's just elucubrating, but I would say that it will be an overlap between the response to virus infection in the lungs, influenza, COVID, or any other respiratory virus, and then it will be, of course, an overlap with bacterial infections. So we can, in that way, define like universal responses to infection versus more virus-specific responses. And if then you compare two viruses of the same family, you can even define which responses are family-specific or even genus-specific or even virus-specific, no? Like levels of responses to infection.
Okay, thank you for the answer. There is another question by Claudio. So he says, we can say that you have a weighted network whose weights change with time. And then he says, if this is the case, have you used homology persistence theory to compare those networks? So,
okay, so yes and no. Yes, they are weighted networks in the sense that by weighted, I assume that you means the nodes in the network, they have a weight, which is the intensity of expression of that particular gene. Okay, so I understand that that's what you mean by weighted networks. So yeah, there are weighted networks that change with time. Absolutely correct. We have never used the homology persistent theory. This is actually, I'm sorry, it's the first time I ever heard that wording. So I don't know what is the homology persistent theory. That would be nice to learn. Actually, I'm going to write down that.
Yeah.
What is that?
Perhaps, Claudio, you can contact with Santiago by email, for example, and discuss this a little bit.
Yeah, absolutely interested.
Okay. So another question by Mark Plana. So he's thanking your talk, which was very nice, and he's saying if you can comment on how does your work on DIPS relate to the ideas you explained today. So this is a huge question.
Hi, Mark. That's a topic for a complete different talk. Okay, the DIs that the project we have started with DIs now, which is very exploratory, and I hope that at some point we will get some bigger money to work on that, and also to complement with the theoretical side together with Josep. It's basically the DIPs. I don't know what you all know what is a DIP. A DIP is basically a piece of the viral genome that is not coding anything, not coding proteins, but only has the signaling for starting replication and ending replication and a small piece of RNA in the middle. So they can replicate much faster than the actual virus because they are shorter and they don't waste energy in translation or nothing like that. So the idea, DIPs were described first time in influenza virus in the mid of the 20th century by von Magnus, and then after that they were discovered in basically every virus. And they are produced during the replication as a waste of replication. Sometimes they are interfering, sometimes not. Sometimes they exist, but they don't do nothing with the virus. But the important ones are those that are interfering. If you have interfering particles, they compete with the virus, they reduce the viral accumulation. Okay, so that's the antiviral effect of DIPs because they reduce viral accumulation. DIPs, they have some other interesting properties. One is that they can be transmitted because they encapsulate as a virus. They can encapsulate together with the virus or independently in a different particle, and they can be transmitted. If they can transmit it by themselves, they don't induce an infection because they lack all the genes. If they're transmitted together with a virus, they will interfere with the virus. So they will kind of stop the spread of the virus. They are very specific of the virus. So you can take the DIPs, flu DIPs, and they will not interfere with coronavirus DIPs. Or you can take coronavirus DIPs and they will never interfere with, I don't know, syncytial respiratory virus. They are very highly specific, and they are highly secure because they have no secondary effects. The only potential secondary effect, and actually it's not that bad, is that because they are viral particles, they can induce an immune response. So they can contribute to trigger the immune response. And in that sense, I never try to correlate or to relate the DIPs work with what I was talking today, but probably we can try to make a connection for the future. I don't know.
Okay, thanks. We have another question answered by David Reguera. So he's saying, usually antiviral therapies aim at blocking the virus. Are you suggesting that it would be interesting to look for drugs that correct the regulation of the network of the patient instead of on killing the virus, or as an alternative to it? This is a very interesting question.
That's another interesting question, and yeah, that's the aim, David, that's the aim. So if you identify what are the targets of the virus and how that deregulates the network, maybe you can somehow rewire the network or add some element to the network that compensates for the deregulation of the virus. And then you will go from a pathogenic interaction to a more permissive or what in plant pathology they call tolerant interaction. Okay, so instead of developing symptoms, you can still be infected, but not necessarily developing symptoms. So that's the idea, to somehow compensate the regulation.
Yeah, at least once the virus has been removed, probably, because yeah.
Or I mean, ideally, to have a sensor in the cells that senses the virus and then automatically expresses something that bypasses the effect of the virus.
Okay. So anybody has another questions? Perhaps some of the other moderators can say something about the talk. If this is not the case, thanks again, Santiago, for this very inspiring talk. This is a very, very novel work that you are developing in plant viruses, but of course, this is a general idea that will be sure be of strong research in the next years.
Yeah.
Okay.
This is a field. Let me just a little bit of publicity. This is a field that really will benefit a lot from the interaction with theoreticians, with complex system people, because as you say, I mean, the references we are using are basically work from a reduced number of theoreticians, of Chinese colleagues, and it would be nice to explore other things. Okay, for instance, this homology persistent theory that Claudio mentioned, I never heard about that before, and I don't know, maybe we can implement it in a simple manner and try to use it. Yeah.
Okay, so thanks, Santiago, again. Thanks, everybody, for coming to the webinar. And now I think Albert is going to tell you something to close this webinar. So I give you the word, Albert.
Okay, just to thank you, Santiago, for this really nice talk and for taking care of all the questions. Thanking also Josep for conducting this webinar. So I know that it's not easy to handle, and then it's difficult to see the reactions of the people. But I think that we enjoyed a lot, and you were saying so. I was checking in the audience, so it's a kind of a mixture of people from the life sciences and more from the physical sciences. But I really think that this is a good opportunity to know exactly what you are doing, as you were saying at the end, what you are doing in these fields, and then at some point being open to establish some type of wide collaboration between the different groups working on this that we nowadays know as this complex system science and interdisciplinary stuff.
Mm-hmm.
Thank you very much. And I just remember for the schedule of the next 2 weeks. So next week we will have Alex Arenas talking about mobility and simulation of epidemic models. And then for the last one, we have another one from Ernesto Estrada. Ernesto is a mathematician, but working also on networks of virus interaction and proteins and all these things. So we will continue on this line. Thank you all of you for being here, and looking forward to meet all of you in a few days again. Okay, thank you very much again, Santiago, and
My pleasure.
At any point, and then when you are able to explain a little bit more about these DIPs, then we will talk again, and perhaps having
we can do a limon
something like this, that we can invite you to share a little bit of our
sure
not just say virtual coffee or a virtual beer. We'll share something else. Okay, you are very welcome. Thank you.
Thank you.
Bye.